You're looking for an honest thing.
We might just be talking to ourselves.
Is there no one in here who looks like
You've got a great choice ahead of you. Well, thanks everyone for coming. We'll get started. So this is just to introduce some really cool topics and research that's going on at IMAS and to hopefully recruit some students to these wonderful research projects. So I'm not going to hold it up too much longer. Joe's going to come and introduce his and Xana's project.
Yeah. I know I've got 2 minutes, so I'll try and be quick. Yeah, I'm Joe. I'm filling in for Xana today, but if you choose this project, you'll be working with me, Xana and George. I'm a PhD student, and we work in a sediment lab here at IMAS. So as a bit of background, my research is towards the paleoclimate and the paleoceanography of the Indian Ocean. This area here between kind of obviously Africa and Australia. And I've already conducted some research in some of these core sites a bit further south, but we've actually got a lot of sediment cores, a lot of material to look at, and some of it's not been looked at all yet. And we're in a really exciting position with this research, because if I go across, we've actually got 3 cores, basically pretty much in the same exact latitudinal position, but at different depths. And because there's a island plateau, you can see that this kind of the depth drops off quite dramatically. So we've got this big depth transect here. The red ones are the ones that my project's looking at, and the black stars are the ones that are new sites that need to be looked at by hopefully an honest student. So our goal really is to look at past paleoproductivity, so past ocean productivity, and also look at and try and understand how past productivity varies with, the export of carbon from that productivity varies with depth. So particularly when we look at changes between the last ice age and the present kind of period, like the Holocene and the recent years, there's ideas and there's research saying that with changing temperature, changing ocean conditions, the amounts of carbon that can be stored in certain areas of the ocean can change, basically. And this is really important to know, because you would have to understand how much carbon can be sequestered into the deep ocean from the atmosphere to help. And also it's really important for the future, because we need to know, well, okay, if we had an ice age that was really cold, we warmed it to the present conditions, this amount of change in the amount of carbon we could take in had, and then obviously we can use that data to predict future what's going to happen when the oceans warm even further. So basically what you'll be doing, sorry, I'm running behind. What you'll be doing is you'll be gaining skills in a variety of topics to paleoclimatology and paleoceanography. So as I mentioned, carbon cycle processes, but also chemical methods, including sediment digestion, chromatographic separation, cannot say that word, elemental quantification. So you'll be using the ICP-MS, or you'll help prepare samples for the ICP-MS down at the CSL, and there'll be analysis of trace metals. So it's quite an intensive lab focus, laboratory focus in this project. You'll get some really good experience, and you'll be working with me quite a lot, because I've already completed a lot of these methods, so I can kind of talk you through it. So there'll be a lot of hands-on there. So it's very much a laboratory-based project, really. And then afterwards, you'll get data quality assurance skills and data visualizations. Obviously, you have to make nice, pretty graphs and tell a nice story about what happened in the past. And then obviously, combine these different data sets. And you'll also be working as part of an international research team with people from America and the UK. So it's pretty cool. And yet, what we hope to achieve, hopefully, I'm running behind, is that you can reconstruct the vertical distribution of water properties, identify how carbon burial sequestration changes with the warming climate, reconstruct past ocean productivity, and also there's a little side project where you can record fingerprints of volcanic activity if you like, because basically I want to just stress that the data you'll get from this project, there won't be tons of samples. Obviously, we know it's only like it's an honest student, you don't have a lot of time, but even in your small data set you'll produce, you'll actually get a load of data out of it, like so much, and you can play with it and tell all sorts of stories with ocean climate history. So it's really cool. Yeah, so that's me. Sorry if I run over a bit, and I'll unplug and let the meeting carry on. All right.
Thanks, Joe. So I think what we'll do is we'll keep all the questions towards the end, and there'll be some food coming, and you can kind of mingle with Joe and pull apart his project, I guess. Yeah.
So Some people are, but some students are online, so, and also this is recorded, so, we find that a lot of people and students sort of watch these recordings later when they're looking for projects.
Next up, we have Elizabeth from CSI Road. Thanks, Elizabeth.
Maybe I should have gone first, Joe. Okay, so this is about the problem. So the problem that faces us all, the problem, you might even say. So over on the left-hand side, sorry, the right-hand side, is I-score data that shows how the concentration of carbon dioxide has varied in the atmosphere over the last 800,000 years. So you see pretty regular oscillations from something like 150, 200 ppm to 250 or 300. This is really at odds with the unprecedented rate of change that we've experienced over the last several hundred years, which is shown over here on the left. So these are observations from Hawaii. In red is the atmospheric concentration of CO2 going up, 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 up. And in blue, you see the oceanic pH, and in green, the oceanic PCO2 or partial pressure of CO2. So what this project is really about is digging into understanding how the ocean is doing us all a great service by tracking or absorbing a lot of CO2 from the atmosphere. So as the red line goes up, 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 you see the green line goes up, up, up. There's seasonal variability, which is captured in the ocean and not in the atmosphere. And importantly, that decline in pH is a direct result of the accumulation of anthropogenic CO2. So the CO2 that comes from our activities releasing CO2 that was not previously there into the atmosphere. And so what this project is about, or the opportunity, if you like, is to use two data sets that are have uniquely been collected about 15 years apart in this area of, will that work? Yeah, so this is the AAPP logo, hopefully shows us Prids Bay. Prids Bay is, oh, I guess that's fun, okay. That's all right, is that working? No, okay, well, so you have 3 big bays in Antarctica around the continent. The size of Prids Bay is somewhat exaggerated in our AAPP logo. Oh, cool, so is this one over here. So Prids Bay, here it is, and there's an Antarctic station there. And the Tempo voyage, this is recently published data by Annie Foppert, who's the primary supervisor for this project, collected observations that you can see in that map there. And long time ago, back in 2006, a very similar voyage was done. And so we have these unique data sets where, unlike what Joe described, the data has already been analyzed in laboratory, so it's ready to go. And the goal here is to be able to quantify how much anthropogenic CO2, so that fossil fuel stuff, has made its way into the ocean in this particular area by comparing 2 data sets. It's really the only way you can do it. You can't measure it directly, and we can talk about that more offline. This is the project team. Apologies to Zana for the blurry photo. It was the best one I could find. You probably all know her and have seen her around. So it's Annie Foppert will be the primary supervisor. She's a research associate with the AAPP program. Zana Chase, who's a professor here at IMAS. Myself, I work over at CSIRO, so it's a joint project between the 2 institutions on the road here. And the last place, of course, is for one of you.
Awesome. Thanks very much. And next up, we have Samantha, who's going to talk about giant crabs.
Hi, everybody. My name is Sam. I work down at the Fisheries and Aquaculture Institute down at Taroona, so I'm not usually here. I'm part of the crustacean research team. And so that's why we have a project on giant crab to put up today. So your supervisory team will be myself. My main focus is rock lobster, so giant crab will be a bit of a new species for me. But Rafa Leon, he is the giant crab expert down in Taroona. And we've also got Stephen Rust, who's a marine economist, who will be able to help out with some of the metrics for the carbon footprint. So sustainability of fisheries is always a bit of a hot topic. Generally, it's focused around the stock itself and exploitation rates and any direct effects such as bycatch or environmental effects such as like habitat degradation, but more and more it's about the entire life cycle of the product. So in this case, giant crab. So the project is going to be using a life cycle assessment to assess the environmental impact of the giant crab fishery in Tasmania and Victoria. So at the moment, there is no assessment for giant crab, any kind of carbon footprint. It's been done for rock lobster, but not for crab. At the moment, we use a proxy based on effort. So basically the number of days that fishers spend fishing for giant crab. That's what this plot here is. You can see that over time, effort has decreased, but basically this is used as a proxy of impact assessment. So more fishing days, there is the expectation there'd be more impact. and with lower fishing days, less impact, but we don't actually know what that impact is. So the objective is to quantify this using a life cycle assessment. This is quite a well-documented approach. It's been used more and more in different fisheries around the world. It's been done for many in Australia. It's been used on the southern rock lobster species, lots of prawns, but not yet for this species. There's a lot of different potentials that you can look at. So this will be kind of up to you to decide what kind of potential impact you'd like to look at. Yes, sorry. Crabs are not usually my my species of research, so I don't really know all that much about crabs. It is a fishery that's concentrated off the northwest of Tasmania. There's a few fishers in Tasmania that operate in the fishery. A lot of them come down from Victoria and fish around in Tassie waters as well. But the drawcards of this is it is very new. It's very needed. We have basically no information. It'll be very important for things like Marine Stewardship Council totally blanked. So certification, thanks Asta. So in the future, this might become very important. Skills gained out of this, there'll be a lot of industry engagement with fishers themselves, processors, trying to find information on where the crabs go to market. You'll be doing surveys, you'll be looking at the LCA approach, and there'll be a little bit of R coding as well. So I think that was very short and sweet, but if anybody has any questions, you can come and have a chat.
Awesome.
Afterwards.
Thanks, Sam. That's great. Next, we've got an online presentation from Simon Goldsworthy from Sardie in South Australia.
Hi everybody, can you hear me there?
I could hear you.
Right, I'll share my screen.
If I can find you.
You might need to allow me to do that.
Yep. All right, Simon.
Give that a go. Can everybody see that?
Looks fantastic.
Hopefully you're looking at a sea lion mother and her pup.
We are.
Okay, terrific. Hi everybody. Yeah, my name's Simon Goldsworthy. I'm a research scientist at the South Australian Research and Development Institute based in Adelaide. And I'm going to quickly give you an overview of 3 projects that come out of a study we've been doing on Australian sea lions using cameras, I guess, to investigate their foraging habitats and feeding rates, diet, and their ecological role. This is a project funded through the National Environment and Science Program. And if anyone picks up this project, I'll be working with myself and Jack Monk, who's not here today, I don't think, as well as Ryan Baring at Flinders University and Gretchen Grammer, who's also at Sardie with me. So why is it important to understand a bit about the benthic habitats and feeding ecology of Australian sea lions? Well, they're Australia's only endemic pinniped, and they're an endangered species. Populations have declined by 40% by 60% over the last 40 years. I should say they're restricted to South Australia and Western Australia, and they occur in sort of 3 main metapopulations you can see there. And yeah, there's not many of them. There's fewer than 3,000 pups born, and they're unusual in that they have an 18-month breeding cycle, so probably fewer than 10,000 animals in total. Populations have declined markedly in the recent decades, and we know we've satellite tracked lots of animals, and we know that their foraging is restricted to the continental shelf waters. And you can see on the bottom there, there's traces from a dive record which shows that the animals travel from the surface to the seabed. They're spending about 80% of their time at the bottom of their dive on the seabed, and they're really just transiting the water column to access air and then get back down to the seabed where they feed again. So the challenge here really is that this is all fine, but you know, if you imagine sea lions were a terrestrial mammal, and you wanted to understand what habitats are important to them and what they eat and some of their other interactions with their environment, you could just step out into a forest or heathland or something and actually see that. But for animals that feed on the bottom of the seabed, right out to the edge of the continental shelf, this is very challenging. And the satellite tracks tell us kind of where they go, but they don't actually tell us what they're doing while they're there. So we've got really limited information about the habitats that are important to them, where those habitats occur, how vulnerable those habitats are to impacts, and the extent to which those important habitats are protected in marine reserves or impacted by other human activities such as fishing. So the 3 projects today, we've actually had a PhD student finish off this, essentially what's been a pilot study, but there's a huge amount of additional areas we want to follow up in, particularly around classifying and mapping sea lion habitats, which will be the first project, assessing the ecological value of different habitats by using sort of feeding rate data and movement and activity data, and then quantifying diet and consumption rates and looking more at the ecological role of sea lions in the different habitats they use on the seabed. So just quickly, this is how we do the work, and there will be opportunities for students involved in these projects to join us in South Australia and assist with deployments. At the moment, we've got 10 deployments we've put out. So we anaesthetize sea lions and glue these instruments to their back. The camera is on the bottom right picture there. On the left-hand side there, that's the camera, and behind that is a archival GPS dive logger that was also linked to a satellite transmitter. We also mount a triaxial accelerometer on the head of the animal, and this can really orientate the animal in 3 dimensions or 4 dimensions under the surface of the water, so we can reconstruct that underwater path in between surface GPS locations. Yeah, as I said, we've deployed 10 instruments at this stage and recorded over 100 hours of video on the seabed, which is massive, really. Over 2,500 dives. So those 10 hours of video footage accounts to over 700 hours filmed of the seabed, which is extraordinary. The animals go to sea for just 2 to 3 days, so we're able to capture quite a bit of their activity while they're out there. So I've got some of the tracks on the right there. You can see in the green of the areas where we've got camera footage, and the blue parts are the tracks where we've only got the movement data without any video footage. So the first project really will utilise the existing data sets and sort of artificial intelligence and machine learning methods to improve the processing of sea lion-borne video data and classify their benthic habitats. Habitat-specific occurrence data will be integrated with other benthic data sets that Jack and others at IMAS have been heavily involved in mapping the seabed off the south of Kangaroo Island and places like that. And we'll be integrating those data here. You can see some of the habitat maps developed from existing data, and we want to integrate those with other data sets that are available. So the aim, as I said, will be to identify and classify habitats on the seabed, integrate with other benthic data sets, and develop improved habitat maps of critical sea lion habitat. Skills for this project will be using artificial intelligence and machine learning to analyse video data, identify the benthic species and habitats and classify those, and undertake benthic habitat mapping. I hope you're enjoying some of the footage being played in the background here. Just gives you a bit of an idea about the kind of imagery we're collecting. So the second project is really about using the movement data and combine, I guess, the video and activity data as a training data set, really to validate foraging activity and prey capture events, which you're seeing here in great detail, and essentially use the machine learning methods to process activity data where we don't have any accompanying video and estimate the foraging activity along the entire track. So one of the important things here, when we've got the cameras on the animals, they dive to the seabed every 5 or 6 minutes, whether they're transiting over an area or actually actively feeding. So the feeding rates that we observe actually provide a really cool way for us to use the animals to value their habitat. So traditionally, us dumb human beings, when we go out and try and value the seabed and identify where marine parks should be, we do it from a very human perspective. We're obsessed by species richness, biodiversity, and habitat structure. And they're the things that we really hone in on when we think we want to protect our marine habitats. But of course, the sea lions don't necessarily care about that. In fact, some of the highest feeding rates we've seen have been on bare sand habitats. So we're getting a very different perspective here. And this approach provides a really unique way to value different habitats within and outside marine reserves. The final project is really looking to, I guess, quantify the diet and feeding of these animals in a lot more detail than we've been able to do. Traditionally, we've been using things like prey DNA extracted from scats to try and get an idea on what the species consume. But it's a very broad approach to doing that. And what we're seeing in the camera data is that individual animals are specialising on different prey species in different habitats, using specialised feeding behaviours to access those food. And we're getting such high numbers of feeding events recorded on cameras. We can actually now use the video footage to identify species, identify their size, and look at the volumetric consumption of different prey over different habitats. And the idea here will be then to build ecological models of what these animals are consuming over particular benthic habitats. And that'll really improve our ability to look at things like commercial fishing activities and the extent to which they may compete with food for Australian sea lions. So that's the 3 projects in a nutshell. Unfortunately, I can't stay to the end of this session, but you can contact me there. My email's at the bottom, simon.goldsworthy@sa.gov.au. And please get in touch with me or Jack if you have any questions. And I'll try and unshare my screen.
That's fantastic, Simon. I think they're going to be really popular projects.
Okay, and just so you can eyeball me, here I am, and enjoy your morning, and please get in touch if you have any further questions. Cheers.
Thanks, Simon. Is there any questions now for Simon in the room or online just before he goes? Nope. We'll get in contact with Simon. I think you're going to be bombarded with emails, Simon, to be
All
perfectly
right,
frank.
see you guys. Bye.
Thanks, mate. All right. Now, I skipped out yarn. It wasn't intentional, I promise. Let me try and get this back up again. Thanks, Jan. Cheers.
Thank you. Cool. Hi. So this is a project between myself and Jack Monk, and this is about underwater imagery and seafloor biodiversity. And the core of this project is really this problem that I've spent quite a bit of time on, looking at seafloor images and extracting the biodiversity information from those images is very time consuming. And we've done that. We've annotated thousands of images from the Antarctic seafloor and extracted what species we could find and how much of the seafloor they cover. But this is very time consuming, and there's tens of thousands of more images that we don't have time to look at. But there's other things we can get from those images that can be extracted very quickly. So you can get all these different types of visual image characteristics. You can batch process, get like, what are the colors that you can see in images? What are the shapes you can see in the images? And the question now is, can we relate those visual image characteristics to that biodiversity information? And it's been shown that that's possible in terrestrial applications, because oftentimes with those image analysis, the terrestrial people are a bit quicker because it's very easy for them. They just take satellite images and then relate that to, and then they just walk along. So it's always a bit easier for the terrestrial people. So they're often a bit ahead in the image space. So yeah, so what we've done is very time consuming, and the other part is very quick. And if we can combine that, we might be able to look at all those thousands of images that no one has time to look at. And by understanding even like a little bit about those relationships, we might then be able to produce biodiversity information, which is not as detailed as this, but which could still be management relevant and we can use. So here's just some examples of different types of habitats and different types of biodiversity information where we get that and how that could look, for example. So in what you'll do in this project is looking at a small data set and annotate that in a time-consuming way, just so you get an idea about the species that occur there, and to generate a little validation data set. And then extract those relevant visual image characteristics from the images, and then look at correlations between the image characteristics and the biodiversity information. So fairly similar, fairly simple correlation analysis. And none of that has ever been done in the Antarctic. So whatever you like, it's a nice frontier. So whatever you touch with Antarctic seafloor biodiversity, usually you're the first one to do it. And yeah, key questions will that relates to will be how reliable are those predictive models then, and how useful can they be, or maybe the relationships are not that good, but maybe we could use that to support artificial intelligence on those annotations. So you'll be involved. You'll get to know a lot about automated image analysis as well. And I think that's it. Thank you.
Thanks, John.
Yeah, my email's down there, so please get in touch.
Thanks. Cool. The next speaker doesn't need a slide. They're going to rely on wit and charm alone. So I think, Stuart, you're up next. The end of emperor penguins.
I think I said rugged good looks. But...
The tissues are up here if you need them.
Thanks, Fraser. Hi, everyone. So, charm, wit, other things. Thanks for coming along. And look, what I wanted to talk about is a bit more about what I really do. And this is an example of a project I could offer next year, but I'm open to sort of talking with students who are interested in projects. What are you doing?
People want to see you online, mate. That's all.
Oh, yeah, stand there. Okay. Okay. So what do I do? I've lectured some of you in the master's program, not so much in undergrad. My main interests are really around how climate change is impacting Southern Ocean ecosystems. So I'm a person who's really interested in the environment and interested in how we categorize change, either using ocean models, climate models, or direct observations from, say, satellite or direct observations, and then trying to understand how that impacts a range of different species. In recent years, I've looked at giant petrels, short-tailed shearwaters, my interest there, doing a couple of things with the southern right whales at the moment, and I'm always interested in Antarctic krill and how we categorise the impact of the environment on krill populations. This particular project is really looking at some work that's been done by people over the last couple of years, saying, well, that all emperor penguins are going to die by the end of the century. The reason for making that sort of statement is looking at fast ice distribution in the Antarctic and looking at changes to fast ice distribution. Apparently, it's all going to disappear, and therefore, emperor penguins that rely on fast ice, which is ice stuck to the continent, to breed means they're not going to be able to breed. Therefore, no more penguins. So this project would really be looking at model output and satellite output to try and identify fast ice and identify when it breaks out, is the term, when it disappears, when it melts. And then correlating that with what we know around habitat use by emperor penguins, where they are using fast ice for breeding grounds. And we can do sort of projections. Projections exist out to the mid-century that might give us an idea of how that would look by 2050 and whether there are significant changes as people are predicting to the availability of that habitat penguin, therefore whether they can complete their breeding cycle. That's all. Happy for questions. And also, if you want to talk about other projects that sort of incorporate use of habitat with impacting on species, happy to have you come and talk to me and we can see what happens. Whose hat's that? That's excellent.
It'll suit you very well.
I think it would.
Thanks, Stuart.
I'll get a tweed jacket.
Our next speaker, Katharina's coming. For the rock jocks amongst you all. Take it away. Thank you.
So hi, I'm Katharina and I mostly work in the geophysics space and also like volcanoes. So, and this one combines the two. Up here you guys see Gaussberg, which is a volcano in Antarctica. The last time it erupted was 50,000 years ago, and it's actually very isolated. So all the other volcanoes in Antarctica are more in Western Antarctica, and Gaussberg is sitting there a little bit lonely. But if you look at that, there might be volcanic activity offshore, and that would be what you guys will be looking at. The supervisory team would include Jodie Fox, who's a volcanologist, who actually stayed on Gaussberg in Antarctica for 3 weeks this year, and Joe Whitaker. So good, there will be good stories from Jodie. Yes, and if we kind of zoom in a little bit around Gaussberg, you already see in the bathymetry there, there's like funny blobs and bobs. And if you look here, this is a reflection seismic line. There are those seamounts breaking out, and they're breaking through the entire sediment column. And while doing that, dragging up the sediments that have been deposited at a certain time. So you can actually date when this happened. And the colored here, those are estimates of the age. So you can actually say that this one erupted quite late in the whole geological time scale that is shown here. So that's really interesting. So the questions you will be answering would be like, where was a volcanic activity offshore East Antarctica? How old was that volcanic activity? Are there any specific time frames? Is there maybe some that they're older in the East than in the West, something like that? And maybe we can look at if they're related maybe to the Kerguelen Plateau or Gaussberg. That's one of the interesting things. There's this giant mantle plume, the Kerguelen plume there, but Gaussberg seems to do its own thing. And kind of the overarching question is, how hot is and was East Antarctica? You will be working with bathymetry, subbottom profiler, seismic reflection data, and maybe some rocks. And you will use geophysical software packages. You learn data analyses on these and use GIS applications. So that would be my first project. And the second one is completely at the other side of the world in the North Atlantic, but there are sediments and there are volcanoes involved as well. So this is a project that is connected to an expedition that I was on last year by the International Ocean Discovery Project 395. So it's a big international effort. There are over 20 scientists working on different questions there, and you will be involved in the whole package, basically. What is happening there? So you all see Iceland, and you all know there's this mantle plume in the volcano below Iceland. Then there is Greenland, which is glaciated, so you have an ice sheet as well. And there are ocean currents, which are super important to the entire ocean circulation on our planet. And what actually happens is that this is the area in the North Atlantic where the deep water is getting produced. So it's an ocean gateway called the Greenland, Iceland, Scotland Ridge, just across here. And there's the cold, dense water from the Arctic coming into the North Atlantic. And they get picked up by this current here. And due to the current, the sediments get reshuffled and build sediment drifts. And those have a very high resolution for paleoceanography questions. And we were looking at the Björn drift, and this is how the sediment package looks like in the reflection seismic. So we have 2 new drill sites. They have been drilled last year, so that's very exciting. The data is not available to anyone else except the science party at the moment. And you would be looking at downhole logging data, so data that has been collected in situ in those boreholes and connected to the sediment that has actually been recovered. So you kind of look at how descriptive are physical parameters to understand the variation in the sedimentation. There are, for example, changes in current speed. So you can see here, or there's also ice rafted debris. And the question is, can we pick this up in the physical data? Then there are also breaks in the recovery where the drill rig just couldn't collect core. And then we kind of want to look at what is actually missing in that succession. You can see that here. This one was not collected and up there where I can't reach. It kind of looks the same. So there's something in the strata that is not recoverable. Yeah, and then we kind of want to look at larger thing, how the sedimentation on Björn Drift is actually react to global general climate changes and the tectonic changes and the pulsation of the Iceland plume. So sediment physical and downhole logging data sets, you would be using, again, geophysical software. You would be working in a large international scientific ocean project and learn skills in data presentation, analytical and writing skills. And if any of those sound interesting, please chat with me, write me an email. I'm happy to have a longer chat on that. Thank you.
Thanks so much. That's cool. Next up, we got Chris. Is he here? There he is. What about productivity?
You guys ever have those bad days where you like sleep in and then you're running late for your lecture, so you rush into work, then you forget to get your bloody parking ticket and you get a parking fine? So sometimes you have these bad days and lots of bad things happen. We start to wonder, is there a shared cause for these bad things or is it just a bad coincidence? So our southeast ecosystems have been having a really bad few decades. And looking across the research at IMAS, I started to notice that there are a lot of bad things going on and no one had sort of put them all together. So we've lost our giant kelp forests, and Scott Bennett's data is showing now that this has resulted in a 40 times loss in productivity of kelp forests on Tassie's east coast. About the same time, we've had urchins invading and eating everything, which, as Scott Link's work showed, has drastically reduced productivity of kelp forests because we don't even have kelp anymore. And then if you talk to the fishery scientists, there have also been a number of different collapses in fisheries. So jackass morwong productivity has declined, and that's not a reef species, that's in the southeast trawl and the shelf systems. There's been productivity shifts in rock lobster, abalone, to name a few. So I was thinking about my bad day and then the bad day for the southeast ecosystems, and you start to wonder, is it a coincidence or is there some shared underlying driver? So this project is to try and bring together all of this information and work that's been done across IMAS into an ecosystem model to ask, is there a cause for this effect? So generally in ecology, we're looking at, we have an effect in mind, like change in temperature or change in productivity, and then we're doing some sort of regression analysis to ask, does that effect cause some change in fish biomass or abalone numbers or whatever? So we're going to use a reverse causal logic. And take all this information from time series we have on kelp forests, coastal reef biodiversity, fisheries information, and bring it together in an ecosystem model to ask, is there a shared cause for all these changes that we're observing in these ecosystems? So yeah, this project will be using modelling, and I love modelling because you can really ask really big picture questions that are hard to ask in the field and lab. So this question spans the whole of the southeast ecosystems and has implications for the way we manage ecosystems for conservation, but also the management of fisheries and understanding how climate is affecting our fisheries in the southeast. So the skills you would learn would be in data analysis and modelling skills that are in high demand by many employers. You get involved with fisheries as well, which is also like an important growth area for employment in Australia. And I know a lot of people might be afraid of modelling, but I'm happy to train. I'm quite experienced in training people in modelling, as long as you have some enthusiasm for getting involved with data and some modelling tools, and I can train you up. So that's about all I have, and I'm happy to chat afterwards. And Scott Bennett on board with this project as well. And then we'd obviously be working across a number of different people within IMAS as well who've talked to and they're keen to be involved and work with us. Thank you.
Maybe a model that could predict the prevalence of bad days.
Yes.
Thanks, Chris. Next up, we have Dale. Dale's in the room. Some ice fish, gonad histology. He's got 2 projects.
I do. And this is the first one, and I'm a fish person, so by far the more interesting to me. So ice fish, for those that don't know, are a subantarctic species. So I work in subantarctic and Antarctic fisheries. The cool thing about them is they have no red blood cells. So you cut them open and it's completely clear, which is fantastic for seeing all of their organs. From a fisheries perspective, maturity is one of the kind of most important parameters that we have for stock assessment models. It's basically when do fish hit puberty and start to reproduce. But for ice fish, which we have about a $12 million fishery for, it's never been looked at in terms of actually looking at their histology. So looking inside their gonads and saying, are we calling what we think is mature, is it actually mature? So this project will look at least 2, but we're hoping to get a 3rd species sampled next year, species of ice fish, and basically look at, do histology. So what that looks like is this. So you take very, very thin sections of the gonads. We work with some of the people at Menzies to use their labs as well to get really, really high resolution images. And it will be basically looking at the developmental stages to say, are these stages what we think they are? And then looking at the general gonad morphology internally, which is something that a lot of people are doing now to then say, what are the different cell types that we're seeing for each egg type? And then develop, at least for one of these species, a model of age-based maturity. So you'll also get to do some otolith aging as well, working at a lab down at the Australian Antarctic Division for that. So that's the first project. It's very quick and simple. It's very much fish biology. If you have an interest in fish or fisheries, it's a good way into looking at that, as well as seeing how the subantarctic fisheries work and working within the AAD fisheries team. The other project, which is very much not fish, but fish adjacent, is we have a project looking at benthic invertebrate interactions with the longline fishery at Heard Island. So when we say benthic invertebrates, in this instance, we mean amphipods and isopods primarily, and some ostracods. So there is actually the potential for 2 different projects here, depending on what your interest is. The first, which is what this one is actually labelled as, is looking at broad scale, kind of categorising the samples into isopod, amphipod, ostracod, potentially going 1 level down below that, and then starting to map those out for where we've seen samples across the Kerguelen Plateau, and then modelling the prevalence of those sea lice against what the fishermen are catching. So the reason for this project is these things come along and they eat the bait off the long lines, which affect your catch rates. They also eat the fish on the long lines, which then affects the value of the product. So we're trying to map out, is there a time of the year or a location that we should avoid in order to stop that? Also, do they have preferences in terms of bait type? The other project which could be done on this would then be, instead of just broad categories, would be, it's a purely taxonomy project of what are the actual species that we're seeing. For this region, a lot of these species have never been looked at before. So if you have an interest in taxonomy, this would potentially be a good one for you. It's working with Kerry Swadling and Luke Brokenshire, who Luke's based down at the Antarctic Division. He works on the CPR data primarily, is a very good taxonomist, and I'm sure all of you know Kerry from various third year classes. She's probably one of the foremost Southern Ocean taxonomists for invertebrates. But that's it. If you have any interest, please flick me an email.
Thanks, Dale. Probably 3 projects here, very easily. Now we're going to move on to macroalgae and seagrass. And I think Jeff's bitten the bullet for this one. You've got 3 to present today, Jeff.
I'm sort of presenting for this one. I'm presenting on behalf of Beth, who's the primary supervisor on this project. So this project's focused on understanding impacts of urbanisation on biodiversity within seagrass meadows, in particular this seagrass species here, Posidonia australis. Tasmania is the most southern limit of that species. Northern Tasmania is the most southern limit for that species. So this project fits within a bigger ARC project, but it's one chunk of it. And I mean, the aims are really to quantify the biodiversity within the seagrass beds compared to nearby unvegetated habitat. Okay, so within the seagrass beds, you've got all this biomass, you've got lots of organisms living on the seagrass leaves or blades, got lots of organisms living in the sediment beneath, in and around the roots. So the prediction would be that there'd be much higher biomass associated with seagrass, but you also find lots of infauna and other organisms that occur in the unvegetated sediment as well. Some of the other context to this project is to sample sites that are being defined as urbanised, so sites that might be impacted by urbanisation, by pollution or some of those factors, and how that affects the biodiversity, and then link the contemporary data to some historical data to try and see if there's any historical changes in biodiversity associated with these beds. So the broader project has fieldwork that goes across WA, South Australia and Tasmania. I'm pretty sure that this project will focus on the Tasmanian samples, or at least the fieldwork component will, but I'm not 100% sure. We'd have to check with Beth on that. So there's some potential to get out into the field. The other part to this is that the sampling will occur before semester starts, before the start of the project. So there would be a need to sort of at least do some of the field work if you're interested in doing that before the project starts. And then after that, there'll be a lot of lab work. So there'll be a whole bunch of samples of sediment, there'll be a bunch of samples of the leaves, and then there'll be a fair bit of time to focusing in on particular taxonomic groups. And the suggestion that we have here is that maybe on bivalves that occur, particularly in the sediment, focusing on bivalves and understanding diversity of those, or focusing on epiphytes. So there's often, it's not too many on this photo, but there's often a lot of macroalgal epiphytes that you get on seagrass. So understanding and quite diverse assemblages as well. So that could be a focus. Right, that's the seagrass project. The other 2 projects I'll talk about are projects focused on this large brown alga here, Davilia, or bull kelp. So for the last couple of years, we've had projects focused on trying to bring bull kelp into cultivation, but at the same time, we've had a number of honours students come in and work on side projects that relate to the broader aim, but we try and pick off things that are interesting sort of side projects for students and to attach to the bigger project. This project is focused on understanding stasis in cultures of Davilia or in zygotes that occur in the lab. So the way we do the cultivation is there's a hatchery stage where you have seed stock. In this case, we have you get reproductive material from male and female plants. You do the fertilization and you create zygotes, which are then grown in the lab for a certain amount of time before taking out into the field to potentially, hopefully, grow bigger. One of the things that's for Davilia that hasn't been done before, but has been done for other kelp, is to work out a way to keep these the really early life cycle stages, so the zygotes, to keep those, essentially keep those in stasis in the lab. And so when you want to grow them, you pull them off the shelf. And you can do that at any time of the year. For Davilia, we can't do that. We're limited to a relatively narrow window when they're reproductive, which is sort of from late autumn into early spring. So 5 months or so. And so that limits when we can do the hatchery stage, and it limits when we can then outplant things into the field. So we want to try and extend both of those. So that's the aim of this project. Wouter, who's here, is leading the project. Fraser's also been brought in for his special skills in so, lots of things. No, applying. So well, maybe I'll get to. So yeah, they're the supervisors. The research problem I've described. The objective is to try and to develop the methods to keep the zygotes in in the hatchery for longer over summer, in particular, so we can outplant in the time of the year when it's a bit cooler. And the way we want to do this is test a number of different methods of trying to keep these zygotes in stasis. One is just keeping them in the dark, in low light or under red light, but in the dark, keeping them at cold temperatures, or keeping them, or putting them in a gel matrix. And this is where Fraser's expertise comes in, because it's a method that comes from microalgae, and we want to try and apply it here for these macroalgae.
That's pretty good.
Yes, so there'll be field work of collecting tissue. There'll be some lab work, which will involve setting up the experiments and culturing the zygotes. So you're learning how to culture these seaweed, and then hopefully getting out into the field and testing how well the more successful ones that have been kept under stasis, whether they can sort of reanimate and grow into large plants once you get out into the field. So that's the second project. The thing about this project is it has to be a semester 2 start. Okay, just because what we're dealing with the reproductive season, that's when we have to start this. The third project is also to do with Davilia, and this is a project where we want to try and develop a molecular method so we can identify the 2 different species of Davilia. So until about 10 years ago, there was only 1 species of Davilia in Tasmania that was split into 2. 2 species are shown up the top there. Their morphology is slightly different, but in the field, it's really difficult to tell them apart. Okay, so often until we get back to the lab, we don't really know which species we're dealing with, or even when we come back to the lab, we're still not sure which species we're dealing with. So we want to develop a molecular tool here. And yes, so that's the problem, is that the objective is to develop this marker. The way we would do that, we're still sort of in discussion about the best method, but we think the way this would happen is there's some genes that we know are variable between the species, and so the idea would be to develop a tool whereby, in this case, targeting the mitochondrial cytochrome oxidase subunit 1, I think it is, the CO1 gene, for both species, which is variable between the species, so the sequences are different between the species. So do a restriction enzyme digestion, and then, if all goes well, then we could run those products out on a gel, and that would separate the 2 species because they have different lengths of those sequences based on the sequence of that gene. Yes, so that's what we want to do. And then if that's successful, ideally, then we'd be able to do some look at the populations we have in the field and to separate, hopefully identify how many of each species there might be at different sites. There's some sites where there's overlap in the species distribution. As far as we know, there's other sites where it doesn't. The data suggests there is an overlap. It's quite likely there'll be hybridization going on here as well, we think. And so we hopefully would have a marker that could help us separate that out as well. That's it. Thank you.
A diverse range of skills needed for those projects, I think. Awesome. We're going to go down south now. Ed's coming up to present his. Let me get the next stuff out. USB-C, eh? Looks pretty good up here.
Yeah,
Hey.
there we go. All right, wonderful. So this is a project about Antarctic bottom water and the timescales of its formation. It's being led by Hannah Dawson, who's a postdoc here in the Oceans and Cryosphere team, and Annie Foppett and I are contributing. So Antarctic bottom water fills up about half the volume of the global ocean, and it's formed in 4 really specific regions around the Antarctic coastline. We know that this bottom water has been changing, and let's go. Okay, that's meant to be a movie. Yep, there we go. Its formation is really intrinsically tied to the sea ice and the water properties on the shelf. We know that its salinity, so how much salt it has, and its temperature have been changing, and we have some observations down from the deep ocean near Antarctica showing us these changes. But we don't know the timescale of the connection from these coastal regions down to the bottom. And we know that what happens is that the water flows off these coastal regions, off the continental shelf. As it goes down the slope, it mixes with the water around it. And the timescale of that transport from up on the coast down into the deep ocean is the target of this project. So Hannah has previously used something called Lagrangian particle tracking, where you put particles in an ocean model and you move them around with the velocities to explore these timescales of connection between places around the Antarctic margin. And so she wants to use that in this project to try and understand the changes in the Antarctic bottom water. So these are some recent papers that have come out about Antarctic bottom water. It's very topical. The changes are really exciting. We think that it's going to be changing even more into the future. And given that it fills up such a huge volume of the ocean, it's really important to try and understand what's going on and how quickly those changes get communicated. So the aims of the project, as I just said, are those timescales. And we're going to be using a model from here in Australia called the Australian Climate and Earth System Simulator. It does a really good job of representing this bottom water formation, much better than other global models. And so you don't need to run that model, but you will get to analyze some of the output from it. So this is potentially quite a data heavy project. Hannah has already developed a lot of the tools and the scripts that you will need to do that. But if you're interested in maybe a slightly more computationally focused project, this is a really good option for you. Thank you.
Thanks, Ed. Last couple of talks now, we have Andy to talk about methane in sea ice cores. Take it away.
Hi students, my name is Andy. I'm sorry I don't have beautiful slides to present today, but I do think I have a very interesting project that I wanted to advertise. This project's about ice core measurements that we are getting online here at the IMAS. And let's pretend for a second this is an ice core from Antarctica. This is a really bad ice core because it's small, for one thing. Most ice cores are about 1 meter to 3 meters long. It's also full of water, which would be really bad if this was a real ice core. But it's about the right shape. So pretend we just took a thin cross section from this ice core and held it up to the light. What you would see is hundreds of tiny air bubbles. The air bubbles are, sometimes we refer to that as the superpower of ice cores because the air bubbles contain a lot of really interesting information. They're almost perfect recorders of the past atmospheric composition. So if we can extract the air bubbles, we can measure things like greenhouse gases, noble gases. We can basically understand in really high accuracy and precision what the past atmospheric composition was. What the Antarctic program is currently doing is trying to extend that beautiful record of CO2 and other greenhouse gases that we saw in the early presentations, tracking something like 8 glacial interglacial cycles back to 800,000 years. We're trying to extend that back to about 1.5 million years. And one of the things that we need to do with this really old record that we're going to retrieve is measure methane concentration in the air bubbles. Why do we want to measure methane concentration? For one, it's a very important greenhouse gas. So studying climate feedbacks in the past related to methane is intrinsically important and interesting. It's also a well-mixed greenhouse gas that allows us to synchronize ice cores between the Northern Hemisphere and the Southern Hemisphere and around Antarctica. So it's a really valuable dating tool. And the last thing is that it tells us some important information about the quality of the gas measurements that we can actually make in air bubbles. What we want to do is build a system. This is what you would be doing. We need to build a system that measures methane concentration in really, really high precision and really high time resolution. The way this can be done is actually by cutting long sticks out of these cores, melting them on a relatively warm surface. And you can imagine that the meltwater and the air bubbles then go into a stream that can be measured in instruments where essentially the stratigraphic continuity of the air bubbles is preserved. So you can measure continuously the methane concentration throughout the length of the core. So what you would be doing is building, testing, and optimizing a system that does this. You would have a blueprint. This wouldn't be like you would be inventing the wheel or something like that. You would have a blueprint for doing this. You'd have close supervision from myself as well as some of the scientists at AAD. And but very much this is a technical sort of, if you like building things, if maybe you played with Legos as a kid or erector sets, if you like tinkering with things, if the idea of being sort of master of your own domain in a lab space and really like being responsible for bringing this online, if that appeals to you, I think this would be a really good project for you. If you want to know more about this, please find me on the 3rd floor of IMAS or hunt down my email. I really should have my email displayed. But yeah, anyway, very cool project, unique, and hope you have more questions and interests.
Thank you. Master your own domain. That sounds great. Last, we have Lauren, who's going to talk about Gould's petrol. Thank you.
Hello, everybody. My name's Lauren. I'm going to describe this project that's happening. It'll be supervised by myself and Jane Younger, who some of you may know. And then there's quite a few other people involved in this project as well, who we can introduce you to if you're interested. So this is a project on an endangered species of seabird, Gould's petrel, this little fellow here. And this is going to be a project that's going to be a 2025 start, and we're looking for a student who has molecular ecology experience. So if you've done that in undergraduate or masters or somewhere else, if you're looking at honours projects at UTAS for semester 2 next year. And why they're interesting is for a very long time, they only existed on 2 populations, basically in the entire world. There's one population on New Caledonia, and then there's another that was sort of in this area here of New South Wales in Cabbage Tree Island. Just over 10 years ago, a couple of these were heard and discovered on a new island, which is Barangaroo Montague Island. It's in New South Wales, but down the very south. And this is very interesting because if you know anything about petrels, they don't get up and change breeding locations very often. This is actually quite a rare event. They're quite a philopatric seabird that tends to come down back to the island that it was born on, picks up, and a new population starts on another island. So we're quite interested to know where these birds have come from. Have they come from Cabbage Tree Island, or have they come from New Caledonia? We're quite interested to know, because these are very, very small populations, because there's been sort of quite a few threats affecting both populations. We want to know about the genetic diversity. We want to know the gene flow, basically who's there, how related to how well related they are to each other, and what's their genetic diversity, how vulnerable are they going to be to pressures. Yeah, there is the potential for fieldwork for this project too, which would be in November, December next year. It won't overlap with Christmas, I promise, but it might come very close to Christmas, just depending on what happens. I can't promise that there'll be fieldwork though, because there's a couple of other factors that we have no control over that might impact that, but we're planning to do fieldwork on both of these islands. Yeah, so if you have a molecular ecology background and think birds are cool, this could be a good project for you. So reach out to myself and Jane and we can answer any of your questions.
Thanks, Lauren. That's great. That's all we have today. Quite a diverse range of projects, and I hope many of you will get something out of today. We've got some food down the front. Dig into it. Otherwise, I'll have to go upstairs to the PhD students. We don't want that. So please help yourself. Ask questions of anybody on any project that you found interesting. All right. Thank you, and thanks to the people online. I think they're still there.
So I don't know, maybe people have questions now. Is there someone? with
Check
questions?
online.
Or people prefer to talk directly?
Anybody online?
Right.
All right, come grab the food and then accost those supervisors. Thank you.